Hello, welcome back. And today we are going to talk about Chapter 18, which is really kind of putting a lot of the knowledge that you've already learned from previous chapters, integrating all that knowledge together to understand pathogenicity. And in this lecture, I'm going to review some key concepts that should sound familiar from previous chapters. And I'm going to give some examples, specific examples of stages or steps of pathogenicity for specific pathogens. But you've already learned a lot through the assignments that I've given previously in this course about the pathogenesis of many bacterial and viral pathogens. So I'm not going to, this is not comprehensive in any way of what I expect you to know, but it's an overview. So remember that pathogenicity is dependent on certain traits and features of microbes, including particular structural features that enable them to carry out the steps necessary to cause disease, so that those structures are related to their function in causing disease. Also, metabolism is driving kind of where they're able to infect a human, what organ system or what site in the body, whether it's aerobic or anaerobic, high salt or low salt, slightly acidic or more neutral. Remember that some of these traits that I'm talking about have been acquired through horizontal gene transfer, or as a result of gene changes or modifications in gene expression. As the microbe enters the human body, a whole new set of genes may be turned on that are important for its virulence and for these steps of pathogenicity. And I'm going to introduce to you some of the ways that pathogens cause damage, mostly focusing on bacterial pathogens, as well as one of the most common ways that bacterial pathogens cause damage in our tissues, hence the symptoms that we have in a disease are through toxins. So I'm going to go over the types of toxins that are found in bacterial pathogens. So you can go over these objectives. These are to guide you in your studying and in your note-taking when reading the chapter. And as you read the chapter, I do want you to think back to some of the features that we've already learned that are important for virulence of bacterial pathogens, or microbial pathogens such as viruses, such as antigenic variation. Okay, so I don't have any specific terms from this chapter for you to learn, but you should be reviewing terms from previous chapters that are relevant to pathogenicity and to these structural and structural features and genetic changes that we've already discussed throughout the semester. So pathogens are different from other microbes that are non-pathogenic, and that's most obviously because they are not capable of causing disease. But why aren't they capable of causing disease? Well, that's because they aren't able to, their traits that they have do not cause some kind of disruption in the normal function of us as a human host. And really I'm focusing on human pathogens and human diseases when I'm giving examples, as we have throughout the course. So these structural features and metabolic features that pathogens have is all based in their genetic composition. So they have a different set of genes or additional set of genes that make them different from microbes that can't cause disease. And they use these unique features that are encoded by these genes to survive basically in the host longer. So how do they survive in the host longer than the non-pathogenic microbes that are cleared before they do any damage to our tissues? Well, basically many of these traits enable them to evade our host immune response. And because they're maybe not indefinitely evade, but at least temporarily kind of delay the detection of their presence or their destruction long enough for them to colonize as part of this process of pathogenesis and cause damage to our tissues. Hence we get sick, we get with the symptoms of a disease, the signs and symptoms. So I just want to remind you that Koch's postulates we already talked about way back in the beginning of the semester. And these postulates, these are a set of steps that are used to link a pathogen to show a causative correlation between a pathogen and a particular set of signs and symptoms that are typical for a disease, for a particular disease. And remember, the microbe has to be found, it has to be isolated, grown away from other microbes, and then introduced into a healthy animal model or susceptible host, and then the same signs and symptoms of that disease must occur in order to show this correlation. And finally, from that experimental model, experimental host, we need to be able to isolate the same microbe. So here are the steps in pathogenesis that are common to certainly bacterial pathogens, but also a variation of which are common to other microbial pathogens such as viruses. So in the case of bacterial pathogens, or any pathogen, first has to enter the organism, right? So we have to be exposed to the pathogen in some way that it gets into our body. Either we breathe it in, or we ingest it, or it lands on our skin and attaches there. Or through secretions or sexual intercourse, the pathogen is somehow entering our body. But once it gets in our body, it can't cause disease unless it attaches and colonizes in the case of bacterial pathogens. And attachment is a prerequisite for colonization. So the microbe won't grow unless it's, in the case of bacterial pathogens, won't grow unless it's able to attach. We already learned about several structural features such as sticky capsule or adhesins that allow this surface, so that are found on the surface of bacteria that allow them to attach well to a tissue or surface within our body, or a specific cell within our cell type within our body that then would allow for them to grow and multiply, which is the step of colonization. During this time, our immune system is detecting these pathogens, and so at the same time that they're attaching and colonizing, bacterial pathogens, in order to be successful in causing disease, must avoid the immune system. Some of these avoid by invading the host tissue, but that's not a prerequisite for all bacterial pathogens. Only some do this. And then once the colonization occurs, there can be toxin production, or the toxin production may occur after invasion of the host tissue. And that's really the most common way that bacterial pathogens are causing damage to our tissues. And then finally, a pathogen will exit our body in an attempt to be transmitted to a new susceptible host and to persist in the environment in that manner. Okay, so this collective set of traits that are encoded by additional genes in pathogenic microbes or pathogenic bacteria are called virulence factors. Virulence factors can be the expression of pili on the surface that help in attachment necessary to happen before colonization. It can be genes encoding a capsule. Remember, we talked about Streptococcus pneumonia, the encapsulated form in the Griffith experiments caused disease, whereas the unencapsulated form did not. Toxins, so even with attachment by something other than pili or capsule and colonization, toxins are included in this set of virulence factors or genes that encode factors needed for host damage or immune avoidance. There are factors that are secreted, protein factors. These proteins can be protein toxins, but they can also be other factors that disrupt normal cellular function in the tissues where the bacterial pathogen is colonizing the human body. Or they may be enzymes that break down components of the immune system, such as proteases that cleave immunoglobulins like IgA, rendering it useless. And so by destroying these key factors that are part of our immune surveillance, the bacterial pathogen buys itself more time to persist in us as a host. And then also we already learned about antibiotic resistance, and so kind of lumped into these. I've lumped survival factors such as being able to resist antibiotic treatment into these virulence factors. So we did learn about plasmids. And within plasmids, these extra chromosomal pieces of DNA that are quite a bit smaller than the genomic or chromosomal DNA, you will find fragments of DNA that are a region within either the chromosome or especially found on bacteriophage viruses within their genome, or within plasmid DNA of bacteria that mark it as different than the rest of the genome. And these genomic islands can have virulence factors, genes that encode virulence factors, and that's when we call them pathogenicity islands. So as we learned before, these factors, if they're on a plasmid or in a bacteriophage, they can be transferred from one type of bacteria to another, or from one bacterial cell within the same species to another that doesn't have this extra chromosomal DNA. And in that way it can spread the ability to cause disease by spreading these virulence factors in a horizontal gene transfer manner. So if bacteria don't have pili and they don't have, if they don't have a capsule, they can still attach by special factors that are proteins located on their pili. So there are adhesins that are on pili, but there are also adhesins that are found on viruses, on the outermost layer, the envelope, that lipid bilayer that some and many human viruses have, or on the cell wall itself if there are no pili in the expressed by the bacteria. And adhesins are these protein factors that are kind of like a key that binds specifically to receptors on host cell tissue, host cells within host tissues, and are able to really mediate a longer connection than just the cell sitting down in a non-specific way on that tissue where it would be easily detached by any movement of the material, the extracellular fluids. So let's look at some of these examples. Pili can be divided into many types. I'm just going to go over type 1 and type 4 pili. The type 1 pili, pili being many, pilus being one, are appendages that are shorter than a flagella, remember, but they're many across the entire surface of the bacterium. And they're used for attachment, and depending on what type of adhesin they have, they bind very specifically to substrates present or present on the host cells. So they're not able to bind to any host cell. It's only which ones the adhesins can make a specific interaction with. Type 4 pili are more flexible. They can move. They can be used not just for attachment, but also for motility in this kind of not the same type of motility as flagella, which give quite a fast directional motility in their run and tumble motility, but unlike flagella, they have many of these type 4 pili. If they're twitching, if they're moving, they're all moving in a uniform manner, in a uniform direction, and so by that uniform extending and retracting, that they're in sync with this extension and retracting, they can move a bacterium slowly through their environment, and this is called twitching motility. They're found both on gram-negative and gram-positive bacteria, but more commonly on gram-negative. These kinds of attachments are important, remember, as the first step in biofilm development. There has to be some way that biofilm forming bacteria attach before they can form a biofilm, either on the surface of a medical device, such as a port, non-shedding surface like that. Remember, much more biofilms form much more readily on non-shedding surfaces, or whether it's within your tissue. So here's, for example, a plastic syringe and the port, and if that port is contaminated with bacteria such as Staphylococcus aureus, which is one type of biofilm forming bacteria, it may be able to attach and form a biofilm, start a biofilm on that surface. And then when it breaks off, it may enter a patient if that device is used to treat a patient. So just to review these steps of biofilm formation, remember that there has to be this attachment, and the flagella is lost when this attachment becomes irreversible. A kind of monolayer of microcolonies forms, but then they start secreting this carbohydrate substance that's very sticky called the exopolysaccharides, and that attracts other bacteria to that area where they can take part in this growth of the towers or the biofilm development, where it grows vertically, and many other types may join at this point. And then finally the biofilm will dissolve and disperse with some of the members of that community of bacteria re-expressing flagella so that they can move to a new site to form a new biofilm. And remember that these are particularly challenging to treat because biofilms are associated with resistance not only to kind of hiding out from our immune cells, but also resistance to antibiotics just naturally by their three-dimensional kind of multi-layer structure and the architecture of that, as well as the components in the exopolysaccharide matrix that would prevent antibiotics and immune factors from entering and neutralize these factors. And so biofilms are associated with chronic infections because of these inherent resistances that they have. So now I'm going to turn my attention to another way that after colonization that damage can occur. The most common way that damage occurs after bacterial pathogens attach and colonize our tissues is by some sort of toxin. And the toxins can fall into one of two categories. There are these exotoxins that are secreted, or even though they're produced in the bacteria, whether it's gram-positive or gram-negative, they're secreted and released from the cell, so they're separate from the bacterial cell. So even if the bacteria dies, they may be released and have persisting effects on our tissues depending on what the exotoxins, what specificity exotoxins have. So some exotoxins are very specific for our nervous tissue and can inhibit neurotransmission in the case of neurotoxins like botulinum neurotoxin. Some are gut toxins, so they disrupt the normal function of our gut in absorbing or reabsorbing water in the formation of the stool, and so they cause diarrhea usually in their effect on our enteric cells such as the gastric or the colonic or intestinal small epithelia of the small intestine. And then there are cytotoxins, which are not maybe specific for other cell types, but they're able to lyse the cell that they're specific for. Again, lysis is a kind of messy way in which cells die, and so this would cause an inflammatory response. So our innate immune system would then be kind of erroneously triggered, and maybe an overblown inflammatory response might be triggered, resulting in not just damage to the pathogen, but also damage to our own tissue. Exotoxins are distinct from endotoxins. Remember, endotoxin are gram-negative, are only found in gram-negative bacteria, and they are not secreted from the bacteria. They're actually part of the bacterial outer membrane, so they're only released when those bacteria die. So let's go back to exotoxins. Importantly, they're made of proteins. This means that they're easily destroyed. So even though they're quite potent and can have fatal damage at very low amounts, which is why an antibody to the toxin is often more effective than antibiotics killing the bacteria themselves. The good news is that even though they're potent, they are protein, and so they can be easily destroyed by heat. So simple boiling or using the autoclave will destroy any exotoxins that are present as a contaminant, along with the bacteria contaminating a fluid or a tool. And because they're proteins, our immune response, our specific adaptive immune response, can generate a specific factors such as antibodies that can bind and recognize these toxins and basically neutralize them before they have a damaging effect, especially in the case of a secondary immune response if exposed more than once to the same pathogen and the same that expresses the same exotoxin. So let's look at a figure from your book that just summarizes how diverse these types of toxins are. Remember, they're all exotoxins, so they're all produced by the bacteria, secreted from the bacterial cell, and they all have some kind of effect on the cells within our own tissues, but different cells within our body, different tissues. So plasma membrane disruption by various toxins will lyse the cell, so these are cytolytic toxins. Cytoskeletal alterations include toxins that will change the shape of a cell, may aid in taking up the bacteria into the cell, and protein synthesis disruption would essentially kill the host cell because it wouldn't be able to carry out the production of the proteins that it needs to survive. Manipulating cell cycle is also an effect, so disrupting the regulation of the DNA necessary for cell cycle, for the host cell to reproduce, or to disrupt some kind of signal that would have a cascade of events intracellularly that would signal for change in behavior of the host cell. If that's blocked or disrupted by these toxins, then can result in an erroneous reaction. So the most dramatic of which is superantigen binding the T cell receptor with in a non-specific way with MHC and activating that a whole number, a much higher number, up to 5% of our T cells, which is way higher than a normal immune response would ever activate, resulting in an overblown immune response that can cause damage to our tissues that we see as signs and symptoms of disease. Cell-cell adhesion could be disrupted, so cells in a tissue are normally interacting with their neighboring cells, but the exfoliative toxin of Staphylococcus breaks these interactions by cleaving the proteins that are holding together these cells, and so you get the loss of the epidermis in things like scalded skin syndrome. Um, and internally, you can have toxins that disrupt the movement of proteins within a cell, and the release of material that from inside the cell by exocytosis. So, for example, the release of calcium by across the synapse is necessary for a nerve signal to be transmitted from one neuron to the next. If the release of neurotransmitters is blocked, that exocytic event is blocked by neurotoxin such as botulinum neurotoxin or tetanus toxin, then the signal is blocked and the nervous system is malfunctioning. And paralysis is the symptom in that case. All right, so let's take a closer look at the subsets. The bacterial exotoxins are divided into different forms. AB toxins are those that have two protein parts. They have an A subunit and B subunit. They might have more than one B subunit. In the case of cholera toxin, it has 5, and Shigella toxin, it has 5 B subunits with just one A subunit. The A subunit is the toxic part, and the B subunit is the part that binds specifically to the host cell that is affected by this toxin. So the B subunit doesn't need to get into the cell. The A subunit does in order to have its toxic effect. In the case of one example of a toxic effect is the ADP ribosyl transferase that is able to inactivate or alter target proteins. In the case of Vibrio cholera and E. coli exotoxins that have the AB form, in the case of Vibrio cholera, this one is again a 5 subunit. Only one of the 5 is shown here in this figure, but it's a 5 subunit, 1 subunit of A, 5 of B. Those B subunits bind specifically to a receptor on the intestinal epithelia. Those villi, or those microvilli that protrude out into the lumen, where the once the Vibrio cholera bacteria have attached and started to colonize, they'll produce this toxin that then binds to the intestinal epithelia, is taken in by endocytosis, where the A subunit is cleaved from the B and released out of that endosome into the cytosol, where it exerts its toxic effect on the balance of the electrolytes by overactivating cyclic AMP, causing a lot of chloride to be released through these ion channels into the lumen of the gut. And then along with that, those chloride ions, there's an imbalance in the substrate concentration across the membrane of these cells, leading to the movement of water through osmosis out of the cell. So a massive movement of water to compensate for that osmotic difference, and that leads to the symptom of very watery diarrhea and massive amounts of diarrhea. And in addition to that, you have the cell death that will occur of these intestinal epithelial cells. And so as they slough off into the lumen and are excreted by in the stool, you see these kind of little white flecks that are the cells in the diarrhea that's called rice water stool. That's typical of the disease cholera. In addition to AB toxins, we have membrane damaging toxins. Those, as I mentioned, will cause, those are cytolytic or cell lysing toxins that disrupt the plasma membrane, like the Staphylococcus alpha toxin that disrupts red blood cell membrane, killing the red blood cell and releasing the iron that it needs in order to grow inside our body. Also includes lysins that affect red blood cells produced by Streptococcus bacteria called streptolysins. Here's a figure from your book just detailing the structure of the alpha toxin. You can see it basically has this kind of tube-like syringe structure on the bottom part of it that pierces a hole in the membrane of the red blood cells. For example, when we did our experiment with blood agar, we saw that a halo would form around the growth of Staphylococcus aureus, and that halo is where the toxin has had its effects. Remember, these are exotoxins. They're secreted from the bacteria into the surrounding area, in this case into the media within the plate, where they are able to poke holes in the plasma membrane, that lipid bilayer of the red blood cell, releasing its contents and causing it to lyse. Those contents are needed for the pathogenesis of Staphylococcus aureus because it needs the iron that's released from those cells to grow and colonize our tissues. The diphtheria toxin is a toxin that disrupts protein synthesis, essentially killing the cell. It's activated by the presence of iron, and it's killing the host cells in an attempt to steal the host cell iron. So it has to, so if there's too much iron, the toxin gene will stop making more toxin. But if there's not enough, it will turn on. So this is an example of gene regulation that's important in the process of pathogenesis that it wouldn't need outside of the host, but it does use this modification of gene expression to express and produce diphtheria toxin when it's needed inside the host. Shiga toxin is found both in Shigella and some forms of E. coli that are enterotoxigenic. So the only forms of E. coli that are damaging to us, that cause disease, things like traveler's disease or hemolytic uremic syndrome, they are toxin producing strains, and the strains that are normally found in our normal microbiota do not produce toxin. So one of those toxins is Shiga toxin that destroys and disrupts host protein synthesis by targeting the ribosome in the eukaryotic form of the ribosome, not the bacterial form. And if that toxin gets to the kidney, it can result in hemolytic uremic syndrome, HUS, which is the lysis of blood cells that are being filtered by the kidney and can lead to kidney failure. And then finally, superantigens are a fourth type of exotoxin that are able to activate T cells in a non-specific way and in a much higher number than would normally be activated. So as high as 20% of T cells would be activated by superantigen, which is way more than the normal level of T cells activated by the presence of bacterial antigen. Hence the name superantigen. It kind of bypasses that necessity that T cell has for activation for binding both the MHC2 and the antigen. Instead, it just requires that the T cell receptor interacts with an empty MHC2, which is found on virtually any antigen presenting cell. Hence the incredibly high percentage of T cells, T helper cells, that are activated by these toxins, leading to an improper T cell immune response. And remember, the helper T cells are also helping to activate the B cells. So both B and T cells would be improperly activated as a result of these helper T cells being activated by a superantigen. Examples of this are the causative agent of strep throat, Streptococcus pyogenes, which has an erythrogenic toxin, and the Staphylococcus aureus enterotoxin, which is the cause of foodborne intoxication. Endotoxin, as I mentioned briefly before, this is that other form. So there are two types of toxins. They're either the exotoxins that we already talked about, those 4 types that are produced, but then secreted from the bacterial cell, whether it's a gram-positive or gram-negative bacteria that produces them. And the endotoxin, it's not found in gram-positive, it's only in gram-negative bacteria, and it's part of that outer membrane. The lipid A portion of the lipopolysaccharide is what endotoxin is. If it's released, it has a toxic effect, but it's a very mild effect, so a lot of endotoxin has to be released in order to result in any kind of physiologic changes. But once a large amount of endotoxin is released, then you get fever, so endogenous pyrogens are being activated. You get clotting factors that are activated improperly, so leading to the slow of the blood flow or blood clots. And then complement is erroneously activated, so just these massive parts of our innate immune response in an improper way are activated, leading to things like a change in the dilation, so a dilation of the blood vessels can result in an opening of vessels that would result in a rapid drop in your blood pressure. Endotoxic shock is usually the last stage before death. So high mortality if the endotoxin is enabled to, if it is left to accumulate to high enough levels, then it can result in death. And this is really only possible when you have bacterial sepsis, so bacterial infection systemically that's infecting your blood. Here's just a reminder of the structure of the lipopolysaccharide, including the O antigen part of the polysaccharide chain in blue, and then the core polysaccharide that has the lipid A at the very end of it. So this would be sticking out of the bacterial cell here on the lipid A end, and the inside of the cell would be over at the O polysaccharide towards, so you'd be getting towards the peptidoglycan layers of the cell there. So as I mentioned, sepsis is what's going to be the only condition that results in the accumulation of these toxins and endotoxins in the blood that would lead to these severe signs and symptoms of toxic shock and organ failure, such as kidney failure and lung damage and impaired gas exchange and improper clotting that blocks capillaries to disrupting the oxygen supply to the tissue. And here's just a summary of the effects of such an gram-negative sepsis due to the release of endotoxin in the bloodstream. So as I mentioned before, complement activated improperly, clotting activated improperly, and macrophages, which then release their soluble messengers in the form of cytokines that lead to all sorts of inflammation and shock. So in the case of endotoxin, it's released when the bacteria die, but when we talk about exotoxins, they have to get out of the bacterium after they're made in order to have their toxic effect. And they can get out through a general secretory pathway that's present both in gram-positive and gram-negative bacteria. But there are some special systems that are some bacteria, especially gram-negative bacteria, use to deliver these potent toxins. Two types that I'm going to go over are type 2 secretion system and type 3 secretion system. So both of these act in a way that allows for the release of the toxin to the outside of the cell in a more specific way. And this secretion is energy dependent, so there has to be an input of energy in order for this to work. In the general secretory pathway, there has to be something that, some kind of channel or something that carries the toxin across the lipid bilayer of the inner and outer membrane of the bacteria, because these are proteins and they're too big to just cross by diffusion. So in the inner membrane, there's a system of proteins that will bind and then carry the toxin across that periplasm to a protein channel in the outer membrane that will then allow for the release of the toxin, depicted here in orange. And that's a type 2 secretion system, where it kind of pushes by movement of these polymers, so additional layers of the polymers being added, it kind of pushes the toxin out through that channel. You can see the polymerization that is a much longer oligomer than you see in the depolymerized state of the protein. Another type of protein that's responsible for secretion of toxins is called the type 3 secretion system, and it's not just toxins but other factors that may affect the host cell metabolism or host cell cytoskeleton that are shuttled through this system. So here we are on the left with the bacteria. It's now produced the protein exotoxins that then, because our protein can't freely cross the inner and outer membranes, but instead it goes through a channel that serves sort of like a needle, where it actually crosses not just the periplasm, but also the space in between the bacterium and the host cell to deliver the exotoxin directly into the host cell. So that's different from the general type 2 secretion system, where it's just released into the extracellular matrix outside of the bacteria, but not directly into another host cell. Okay, so I'll finish up with this part about other ways that pathogens avoid host defense next time.